This video will focus on genomes and genomics, and some of the broad categories of topics that we'll focus on are listed up on the screen. And let's jump right in and begin our discussion with DNA sequencing. What we're going to do is discuss the way in which DNA sequencing was first worked out back in the 1970s. It's called the Sanger sequencing method after the person who figured out this basic process, and it's really quite brilliant how it works. And it relies on having these modified bases, namely dideoxynucleotides. And essentially they're illustrated here. These are bases, these are essentially modified A's, T's, G's, and C's, that when they are incorporated into a newly synthesized piece of DNA, they stop DNA synthesis at that point. And that's crucial for the process of Sanger DNA sequencing. So how it works is like this. You take a sample DNA, which will serve as a template. You add all of your normal bases, A's, T's, G's, and C's. You then, of course, will need a DNA polymerase, and you need a primer, so a short sequence, in this case, of DNA. That will serve as a primer, and it's typically labeled in some fashion or another, typically with some radioactivity. So now you add all these together, essentially have everything you need to do DNA synthesis in a test tube. But now you separate all of your components into 4 separate reaction mixtures, and to each of those you'll add a separate dideoxynucleotide, so a separate dideoxy A, T, G, or C, again into one of each of 4 tubes. Then what essentially happens in each tube is illustrated here. This is illustrating what's happening in a tube with a dideoxy A, and what we're going to do is start at the very top. Notice, this is from your textbook, notice that they put the 5' on the left and 3' on the right for the original template DNA strand from the sample. Perfectly fine, perfectly okay. Just recognize that in class often I will flip the piece of sample DNA, so I'll put the 3' on the left, the 5' on the right, and the only reason is it's just personal preference. I prefer to have DNA synthesis occurring left to right, but here it's occurring right to left. Again, doesn't matter, same result, but just keep that in mind. So again, here we have our sample DNA. It contains a sequence. We don't know what that sequence is, but our goal is to figure out what that sequence is. So what we have on the far right is our primer. This is our short stretch of DNA that we introduced into our reaction mixtures. Our primer finds a region that's complementary to our template. It binds there, and again, it's labeled with some radioactivity. And then imagine newly synthesized DNA being formed. Of course, every base that's complementary will be added, so a G here, a C, T, T, A, and so forth. So if we jump down here again, that's what we just discussed. But now notice at this location, we're going to introduce an A. If that's a dideoxy A, that will stop DNA synthesis at that point. Right below it, if it happens to be a normal A that's incorporated, DNA synthesis will continue, again in this case right to left, until we encounter another spot where an A will be introduced. Again, if it's a dideoxy that's incorporated, DNA synthesis will stop. If it's normal A, it will continue. So in this particular tube, again with the dideoxy A that was added, we're going to form all possible lengths of DNA, each of which terminates in an A. So think about that same logic for all of the other 3 tubes with the dideoxy T's, G's, and C's. And what you're going to form among all of your 4 tubes are essentially all the pieces of DNA of different lengths that consist of the primer, and let's just say the primer is the light gray, so the primer sequence is TAG. So the primer plus 1 additional base. So primer plus 1 base here, in this case it terminates in a G. 2 additional bases, in this case it terminates in a C. Next one terminates in a T, next one a T, and so forth. So again, these would be actually kept separate amongst your 4 tubes. Here I've just sort of combined them together. But now imagine when you run these out on a gel, again keeping your 4 lanes, your 4 tubes separate here, each one corresponding to the dideoxy that was added, so dideoxy T, A, G, and C. And how you would read this gel, again recall that the smallest pieces of DNA will zip right down to the bottom. The larger pieces of DNA will stay behind more toward the top. How you read this gel? You essentially start from the bottom, work your way up, and just keep track of your bases directly. So G, C, T, T, A, G, and so forth. So what we're doing there is we're essentially keeping track of the sequence of bases directly from the gel. So at that point, the base toward the bottom, that will represent the 5' end of the sequence that is complementary to our original sample DNA. So again, if the sequence directly from the gel, the bottom would be 5'. So 5' and then G, C, T, T, A. But again, that's the complement to our original sample. So what you would do next is then take the complement of that sequence, which would be, in this case, again, don't forget you have your primer initially, but the first base after the primer would be, again, the complement would be C, G, A, T, C, T, A, and so forth. In that case, the 3' end is at the bottom. So again, that's just important to remember. So again, the original, when you're reading and converting to your original sequence, when you're dealing with the original sequence, after you've already found the complement to what's on the gel, again, the 3' would be at the bottom. So that gives you basically your sequence of your sample. So that would be illustrated here. Again, 3' on the left, or corresponding to the bottom. You have your primer, the complement to your primer shown here, and then C, G, A, T, and so forth. So that's how you figure out the sequence of bases in a DNA sample. The reality is these days, people just don't do that anymore. It just takes too much time. It's too expensive. So you might have heard of next generation sequencing. We won't talk about the details here, but in a nutshell, this is a much faster, far more inexpensive way to sequence DNA. And instead of relying on dideoxynucleotides to terminate DNA synthesis, it simply relies on techniques whereby a flash of light is emitted every time a base is incorporated, and so computers can keep track of that and generate a DNA sequence far more rapidly than you can with Sanger DNA sequencing methods. A basic concept that you might come across is the idea of whole genome shotgun sequencing. Genomes can be quite large, so how would you go about attempting to sequence an entire genome? Well, in a nutshell, what the approach that people tend to do is essentially, I'll just jump to the next slide here, is essentially take the genome and cut it into many small pieces. You can do it using enzymes or various physical methods and so forth. But cut into many small pieces, you can then make clones of each of those small pieces. Then you can sequence each clone. So each sequence at this point will be a fairly small sequence. But then, of course, using computers, you can look for regions where those sequences overlap, like we see illustrated here in step 4. When you get these regions where sequences overlap, you can then organize those into what we call contigs, for contiguous pieces of DNA. Then you can look for overlaps among your contigs, essentially to generate an entire genomic sequence. So obviously this can take a lot of time, but it's much more doable now with next generation sequencing techniques. I'll just quickly mention that there are some public databases that anyone and everyone can use at any point. GenBank, for example, maintains a lot of gene sequence information, and you can also go and use the BLAST website. BLAST refers to Basic Local Alignment Search Tool. This would just be a website that you could use if you had a DNA sequence or an RNA sequence or even an amino acid sequence of proteins. You can sort of plug those in to see what gene you might be dealing with. I'll just point out here that there are a variety of fields of study that are burgeoning these days. For example, bioinformatics, very broad term here, refers to basically the study of genomes and the information in genomes. If you imagine, if you think about genes, right, you've got exons, right, that of course is sort of the protein encoding portion of genes, but there are all sorts of other regulatory regions that come into play as well. So bioinformatics really is aimed at understanding not only the protein coding regions, but all the other regulatory regions among genes and how they interact with each other as well. There's also a sort of a subfield, if you will, called comparative genomics. What we're talking about here is a field of study where we're comparing genomes across different species. So we can look at humans versus chimps, even versus Neanderthals. We can isolate Neanderthal DNA and do studies with it. And so some interesting things start to occur. We can use these sequences to construct phylogenetic trees. and look at evolutionary relationships among species. We can also look at genes, gene structure and gene sequences among different species. And so, for example, what's illustrated here, you can imagine chickens, they've got a variety of genes that are involved in producing yolk. So here we're illustrating three yolk genes found in chickens. And it turns out that humans and even other mammals, here's a platypus here, it's an egg-laying mammal, actually have either those genes or remnants of those genes. So the platypus, if we take a look down here, it still has yolk gene 1. That's functionally active. It also has remnants of the other two genes as well. They're still there. They're still recognizable as the remnants of the yolk genes we find in chicken, but they're not functional. So they might have the same regulatory sequences and largely the same protein encoding sequences as the functional yolk genes, but again, they're modified, they've been mutated over the years, and so they're simply no longer functional at this point, so we recognize those as pseudogenes at this point. Humans, we have remnants of the yolk genes found in chickens. Again, they're not functional at this point, but again, their sequence is close enough that we still recognize those as corresponding to the genes found in other animals. But again, we would say at this point they're considered pseudogenes simply because they're not functional. They don't code for functional protein products. And I also would point out a couple of concepts here. We can talk about homologous genes. Here we're talking about genes that have been inherited from some common ancestor. So we can talk about this in two different ways. So on one hand, we can say, well, the yolk genes in the chicken and the human and the monotreme here, the platypus, they represent orthologs. So here we're talking about, again, homologous genes that have been inherited from a common ancestor, and they essentially have similar functions across the different species. Even though I recognize at this point that those genes are not particularly functional in humans and monotremes, but the point is they were inherited from a common ancestor, and those same genes are found in, for example, these three species. So we can say, well, those are examples of orthologs, but we can also talk about paralogs. So what I'm not illustrating the details here, but you see these three yolk genes, either in chicken, humans, or monotremes. It turns out there's good evidence that they were formed from gene duplication events. So what can happen by a variety of mechanisms is genes or large sections of DNA can be duplicated. So initially there might have just been one yolk gene that could have then been duplicated once or twice or a dozen or so times. It depends on the gene in question. So they end up forming multiple copies of the gene in the organism. So in that case, where you have genes that are formed from gene duplication events within a species, again, you could still say they're homologous, but in this case, you would say, well, they're examples of paralogs. Okay, so that's just the difference between orthologs and paralogs. Also illustrated here again, orthologs, same genes across species. Paralogs, shown in yellow toward the right. If you have a gene that's duplicated, again, now you have two genes. You should say, well, they're homologous, but in this case, they're within a species, so we say they're examples of paralogs. We can also briefly introduce this concept of synteny or syntenic DNA regions. The basic idea here is if you look not only at the sequence of bases in genes, but also the order of genes across species, turns out often there's lots of similarity among the chromosomes. So here, this figure is confusing, but basically you're looking at a chromosome from mouse toward the top, a chromosome from humans toward the bottom, and all the regions in purple represent syntenic regions. In other words, large sections of DNA that contain lots of linked genes that are found in the same order. So again, not just another evidence of DNA being inherited from some common ancestor. Now let's turn briefly to this field of functional genomics. Essentially, we're talking about gene expression patterns. So the products of gene expression, how they interact with each other, and how they interact with DNA itself. This is a whole field we can refer to as functional genomics. So we can talk about the transcriptome. This essentially would be all the mRNAs produced within cells. Or we can talk about the proteome, which of course includes all the proteins produced within cells. And so now let's just take a few seconds here and discuss comparative gene expression. So, for example, you might be interested in a variety of questions, one of which might be what genes are expressed as organisms age, for example. So you could look at many different species, and you can use techniques to see how gene expression changes in each of those, and then do a comparison among those species to see if there are any similarities or differences. So what we're going to do is just take a brief look at a technique that we can do to study those and address those types of questions. But first of all, let's just introduce some very broad basic concepts here. First of all, this idea of reverse genetics. This is just one way in which people can address the relationship between genotype and phenotype. So in a nutshell, reverse genetics is simply the approach whereby if you find a gene of interest, but let's say at this point you have no idea what the gene really does. So through reverse genetics, you can mutate that gene, and there are a variety of ways to do that, but then look to see what impact that has on the phenotype of the organism. That will then give you a clue of what that gene actually does. On the other hand, you might take a forward genetic approach, and in a nutshell, it goes like this. Think of some phenotype that you're interested in. It could be aging, it could be obesity, it could be just about anything you're interested in. So find a phenotype that you're curious about finding the genes that play crucial roles in that phenotype. So what you can then do essentially is take some test organisms. It could be mice or it could be fruit flies. Let's just say fruit flies, for example. You can then introduce mutations, random mutations in your organisms. So in this case, in your fruit flies. You can do that in a variety of ways, chemicals, radiation, and so forth. And then you can look to see, you can screen your population of fruit flies to see which of those show the changes to the phenotype that you're interested in. So if you're interested in longevity, right, or old age, you can look to see, okay, which of those mutated flies either live very long or perhaps die young, to see what role those genes might play, again, in living a long time and reaching old age. So the point here in forward genetics is you're starting with a phenotype in question, and then you're sort of looking to see what genes are involved in determining that particular phenotype. Okay. So how can we then approach this, any question dealing with differential gene expression? There's a basic technique called microarrays that was developed a decade or so ago. In a nutshell, if you look at the results of a microarray analysis, you might see something like we see here. What are these? How are they produced? And how do you interpret these sorts of things? Well, in a nutshell, it involves producing so-called microarrays, which typically are just sequences of DNA attached to glass slides. So these are sequences that represent genes of an organism, and they are known because people create the microarray slides, like we see illustrated here to the right. So we can print or attach specific genes to this glass slide at specific regions. So again, we know which gene is where and so forth. We keep track of these. But then what we can do is take this microarray, this glass slide right here, and then inoculate that or introduce that to CDNA from tissues of various types. So for example, first of all, actually, let me just quickly remind you that assuming no problems with color blindness, we can see green, we can see red. When we stimulate our green and red cones together, we perceive that as yellow. And the reason I mention that is because understanding that is crucial to understanding how microarray analysis works. So again, keep that in mind over the next few minutes or so. So again, how does this work? So imagine we've got two types of cells. It could be a control cell and a cancerous cell. It could be, you name it, liver cells versus brain cells, whatever you're interested in. So let's just say we have control cells on the left, treated cells on the right. Perhaps these are cells treated with some drug or a poison, doesn't matter. We can then look to see how gene expression is different among these two cells. And it goes, again, using microarrays, and it goes something like this. So let's say that these are cells from the same species, so they've got 5 genes here, but of course not all the same genes are likely to be expressed in these two conditions. So cell on the left, control, we have protein 1 and protein 5 produced. On the right, we have protein 1 and protein 4 produced. So what we can do as our first step is isolate all of the RNA, all the messenger RNA from those two cell types. We can then convert that to CDNA using reverse transcriptase. On the cell on the left, however, we're going to include a green fluorescent molecule as we produce our cDNAs. On the right, we'll add a red fluorescent molecule, again, as we produce our cDNAs. So now we have our cDNAs from these two cells. We can then add them to our microarray. So we have them in a test tube here. We can literally add them to our microarray. Again, this represents the glass slide, right, with the genes printed in specific locations. So in simplified format here, let's say we just have 5 genes that were printed on this microarray, corresponding to the 5 genes that we had illustrated initially in our two cells. What's going to happen is that if you have a cDNA that's complementary to a gene on the microarray, they will hybridize, they will bind. Okay? So what's going to happen for DNA 1, if you recall, both the control and the experimental cell express gene 1, so you'll have cDNAs from both of those cells hybridizing to this spot. Neither of the cells express gene 2 or gene 3, so you have no cDNAs there. One of the cells, let's see, it was a cell on the right, expressed gene 4, so you'll have cDNA from that cell binding to this spot. The cell on the left expressed gene 5, so you'll have that cDNA binding to this spot. So essentially what you can do then is you have your microarray, you then scan it with lasers to check for fluorescence, and when you scan it initially, let's say you're checking for green fluorescence, you'll have spot 1 and spot 5 fluorescing. When you scan it for red fluorescence, you'll have spot 1 and spot 4 fluorescing. You combine those two to get a single readout, and you get something like this. Spot 1 appears yellow. Why? Well, again, because both green and red fluorescence will appear yellow. And that indicates, if you see a yellow spot, that indicates that that gene is expressed in both of your two types of cells. On the other hand, just one of the cells expressed gene 4, and just one of the cells expressed gene 5. So again, those will be either red or green. Spots 2 and 3 are blank, indicating that neither of the cells expressed those two genes. So in a nutshell, that summarizes how microarray analysis works, and again, how it can be used to study differential gene expression. That concludes our brief discussion of genomes and genomics.